or becomes is the result of the nucleus, because in the nucleus is our DNA, our genetics. Okay? So as we look at a nucleus, I believe I mentioned this before, that it's a double bilayer membrane. Okay? So really it's 4 layers. A double bilayer of phospholipids. And it has these holes in them, and these holes are called nuclear pores. You can kind of see them all over out here. You can kind of see that the double membrane of the nucleus just comes on out and makes an extension here, which becomes the endoplasmic reticulum with the ribosome studded on there. But as we look inside the nucleus, we kind of see this blurry, fuzzy-looking material. I don't know how to describe that other than that. Granular-looking. This is called chromatin. Okay? And you may even hear it called euchromatin, which means true chromatin. What chromatin is dispersed genetic material. It's your chromosomes all relaxed. Okay? Not getting busy, not doing anything, just chilling. Okay? When we start to organize this chromatin, and we start to visualize it, we can actually see it start to form into the characteristic X that we recognize as being chromosomes. We call that heterochromatin, or chromosomes. Okay? Well, there's only one time when that will take place, and that is when we're getting ready to divide the nucleus. Okay? And we'll talk about the difference between nuclear division and cellular division. Because probably all your life you've been told that cell division is mitosis. Most of you will probably recognize that term as being taking one cell and making 2 identical cells. But in reality, mitosis is simply the division of the nucleus. Cytokinesis is the division of the cell, okay, which happens in the latter phase of the whole process of cellular division. So we're going to kind of bump up what you know and just add to it a little bit. Okay? Well, this also has within the nucleus a nucleolus, which is just condensed, very well-organized nuclear material. Okay? And so, kind of like planet Earth has a core, right? Yeah? Why shouldn't our nucleus have a core? Because when you think about it, this is just a little Earth. Okay? And when it divides, it's an earthquake. Does not make sense to you? No? Okay. Well, anyway, be familiar again with these structures. More especially, look at this graphic down here. Okay, this is magnified 10,000 times on an electron microscope. And this is the nucleus, right here. All of this would be the chromatin. This is the nucleolus. Has a nuclear envelope. Okay, and then it has out here the endoplasmic reticulum. Okay? Ladies, I need you to stop this, or take outside one or the other. It's not bothering me, and it's bothering everybody else. Okay? So, make sure you're able to identify the structures of the nucleus. Alright? Alright, function. It shouldn't be a big surprise to you. Stored genetic material. This is where your DNA is. This is where what you are, what you look like, your hair color, your shape of your nose, your face, your eyes, your ears, the color of your skin, all of that is programmed right here inside the nucleus. Okay? And so, we know that DNA is deoxyribonucleic acid. We learned that last time. Here I'm talking about the loose chromatin, known as euchromatin. And package type, heterochromatin or chromosomes. Okay? When we get ready to make a protein, we're only going to take half of a chromosome, copy it, and then we have an RNA. And the RNA is going to come out of the nucleus, attach to the ribosome, and it codes for nucleic acid. It's going to come and attach itself, making peptide bonds, actually making hydrogen bonds for the secondary level, disulfide bonds for the tertiary level, and then a multitude of all those bonds for the quaternary level, and then we have a protein, being 5,000 or more amino acids, making a protein. Okay? Well, that's what's going to take place both inside the nucleus and outside the nucleus. The one thing you need to understand, DNA can't leave the nucleus. RNA leaves the nucleus. Okay? How does it leave the nucleus? Through the pores. Okay? Now, anybody familiar with the term the central dogma? Somebody know what that means? This is a term that was coined probably back in the 13th, 14th century. Okay, anybody know what the current affairs of Europe was, Eurasia during this time? What type of scientific discoveries were going on? 13th, 14th century. I will tell you, none. This is the dark and Middle Ages. This is when everything's dead. Okay? It wasn't until about 1492 when we started to have some discovery, because one man got really brave and challenged the church. That would be Christopher Columbus, for those of you that know about 1492. Okay? Well, during this time, there was the term that was used to describe truth. And it was the central dogma. Okay? And you did not question it. Okay? Because if you did, what happened?
You're dead.
Yeah. Any number of things could happen. Okay? If you were lucky, they just cut your head off. Most got disemboweled. Sit there and pull your guts out while you're still alive, and you're just having all that take place. Okay? So on any number of things, if you went against what was the central dogma. Well, then we come along with discovery. We move into the Renaissance. We move into what we now know as the 18th, 19th, 20th century, that explosion of learning. And the central dogma changed. Okay? Because what was true in the 14th century was that the planet was flat. Columbus pretty much put that to bed. Okay? And those that followed him began to change the truth. Okay? And now all of a sudden, we can't have a central dogma. What do we have to have? We have to what's known as a flow of information. And so the central dogma over the centuries has changed from truth to flow of information. What that means is we're going to take what you know, we're going to add to that, and it might change what you believe to be true. But you got to be okay with that. Okay? Do any of you know anybody that are from the Flat Earth Society? Okay, a few years, well, many years ago, we actually paid somebody to come here to Weber City to speak in a convocations who was of the Flat Earth Society. And if you challenge this individual, we got this going on in the convocation, you challenge this individual with what we know to be fact, he says it doesn't exist. So someone says, well, do rocket ships leave the planet? Yeah. And are they taking pictures of the planet? Yeah. Well, don't those pictures show that there's a horizon and it's round? That's not true. You can't argue with that logic. Okay? You can't. If they're unwilling to accept that their paradigm has to shift, then they're going to stay in their ignorance. Okay? Well, we have adopted the central dogma in anatomy and physiology to say, look, today this is true. But we're going to take that information, we're going to build on it, and tomorrow it may not be the same truth, but it's still going to be true. We've just added to it. Okay? And so we call that now the flow of information. And I'm sorry for the tirade here on the explanation of the central dogma, but I think it's important to understand that even what I'm telling you right now could change. Why?
Because science, because they find new things out all the time.
Yeah, new technology could come up that actually says, no, that really doesn't happen that way. Pretty close, but yeah, not close enough. So we have to change our way of thinking, okay? Anybody know who Mendelian is? Not Mendelian. Does anyone know who Mendel is? Excuse me, who Mendel is? Gregor Mendel, anybody? You need to go slap some science teachers in your high school.
I need to come up with a periodic table.
No, not periodic. Gregor Mendel was a what? No. Gregor Mendel was a monk who lived right around 1400, 1450, whose job it was to grow garden vegetables so the monks would have something to eat. Okay? Gregor Mendel. He grew peas. What did he find out about these peas? They grew different colored flowers, and that blew his mind. He, wait a minute. Same pea out of the same pod, different colored flowers. How does that happen? Well, he's the first one to come up with theory of genetics. We actually use Mendelian genetics today as the foundation of what we know to be about genetics now. Okay, so everything you're going to learn tonight about DNA, Mendel theorized it, but couldn't see it because he didn't have the technology. It wasn't until Anton van Leeuwenhoek came along and invented the microscope that we were able to see some of the things we're able to see now. Okay, so I don't know. Are you guys really not interested in history at all? Does history just bore you to death? You know, it used to bore me. And U.S. history, no use for it. European history, no use for it. Medical history, I like medical history because now I understand why I'm doing what I'm doing. So, yeah, take a little bit of time, read a little bit of medical history, it's okay. Okay? Enlighten yourself.